and welcome. I am trying to learn cytology today, transport across the plasma membrane, and after this download we're gonna do leg injury prevention training. And if you participate in the leg injury prevention training, then it's the second time that we're reading it. Transport across the plasma membrane. Transport of materials across the plasma membrane is essential to the life of a cell. Certain substances must move into the cell to support metabolic reactions. Others must be moved out because they have been produced by the cell for export or are cellular waste products. Some things have to come in so that the cell can do metabolic reactions. Some things should go out because they are wasted or they were produced and should be delivered. As you have seen, some substances can cross the lipid bilayer portion of the plasma membrane, whereas others use channels or transporters formed by membrane proteins. So I think we're going to save it. We're preparing a little presentation. We're going to do not the presentation so fast. We're going to make probably first the other ones all. But I think then we're going to say a thing here about water. The lipid bilayer portion of the membrane is permeable to most nonpolar uncharged molecules such as oxygen, carbon dioxide, and steroid, but is impermeable to ions and charged or polar molecules such as glucose. So oxygen, carbon dioxide, and steroids can go like wet through, I think is what they say here. If they say it somewhere else, more clearly. Maybe. But then water, for example, can go through. As the fatty acid tails of membrane, phospholipids and glycolipids randomly move about, some gaps briefly appear in the hydrophobic environment of the membrane's interior. Water molecules are small enough to move from one gap to another until they have crossed the membrane. So water have here at least. So water as a thing going through it, as an example, and then we have channels and transporters. Channels letting when run a specific substance through, a transporter taking a specific substance and then bringing it through, shaping its, for changing its shape in. Receptors are taking a specific substance and that is what we're talking about in the next chapter. So channels and transporters. Okay. Substances cross cellular membranes via transport processes that are classified according to two criteria. One, whether they are mediated or non-mediated, and second, whether they are active or passive. In mediated transport, materials move through the membrane with the assistance of transporter protein, whereas in non-mediated transport, no transporter protein is involved. So that was when, I don't really know where it was actually described. They had when here facilitated diffusion, which should rather be called, do they say, facilitated transport. So it was somewhere here written, but I think it know what it was. It is coming through with another thing. Where was it? Here was it. So one substance goes out and through that another can go in. But it was it active transport? We're going to go to that all in the next part. But do we have what we want to have here? So we want to have that. Mediated, non-mediated. What is no difference and what was it exactly? And second, active and passive transport. So active adenosine triglyceride is used to go to take the phosphate, I think, the ATP into ADP, a reaction that also attaches a phosphate group to the pump protein, and when the pump is changing, the calcium is going out and the potassium is going in. And the passive secondary active transport, that was that through it going in and out, another one can go out because another one came in or something. We're going to go there all in detail. But we know what we want to look for. In passive transport, a substance moves down its concentration gradient across the membrane using only its own kinetic energy of motion, whereas in active transport, cellular energy, usually in the form of ATP, is used to drive the substance uphill against its concentration gradient. So passive, it goes there because there's less, it goes to the place where there's less. By active is it help to get even more over even when there is already more. Living cells use 3 passive transport processes. 2 of these are non-mediated. Diffusion through the lipid bilayer and diffusion through a channel. Okay, these are the two we just had. I think that was, for example, the water is going through, and the other one going through the channel. They can go something through the channel. One passive process, facilitated diffusion, is mediated. Yes, it is mediated. Because we said here, it should maybe not call facilitated transport. A solute binds to a specific transport on one side a solute binds to a specific transporter on one side of the membrane and is released on the other side after the transporter undergoes a conformational change. One process, facilitated diffusion, is mediated. Among active transport processes, all of which are mediated, we can distinguish several types of transporters. Uniporters move only a single substance across the membrane. In coupled transporter, two substances move across the membrane together. Coupled transporters are either symporters, which move two substances in the same direction, or interporters, which move two substances in one direction across the membrane. In opposite directions. Symporters, here two substances to the same side. Interporters, one out, one in. Another way that materials may enter or leave cells is by processes that involve the formation of the membrane surrounding vesicles. In vesicular transport, tiny vesicles either detach from the plasma membrane by bringing materials into the cell, a process called endocytosis, or merge with the plasma membrane to release materials from the cell, a process called exocytosis. Large particles such as whole bacteria and red blood cells and macromolecules such as polysaccharides and proteins may enter and leave cells by vesicular transport. We read it in the previous part. So that goes to it, when it pinches off, goes on around, comes together with an early endosmer with other carrier vesicles. It can go on as a carrier vesicle and sort back to the plasma membrane. There was written how cell can do its whole amount of plasma membrane every 90 minutes. Or it can go on to form lysosome and be then ready to do something, whatever. Endocytosis means in, exocytosis out of a cell. And there can be phagocytosis. What was where it goes like that around and takes it in with the white blood cells that we're getting bacteria in when other carrier vesicles come to it and destroy it when inside of them. And then there was pinocytosis, what was that? Where it goes not, where it does not go to it. So first was vesicular transport, it goes to it and then pinches off with a clavulin coated plate. When phagocytosis was, it takes it, but then it goes around it and takes it then in. And pinocytosis, it isn't something binding to it, it just goes in and takes something in it. But therefore was necessary. I don't know if I ever said what the use of it is. In most cases, the residual body fuses with the plasma membrane and expels its contents from the cell via exocytosis. Yes, and therefore was it what? And then the exocytosis to be outside. Okay, now we have it all together. That is what we want to have. That is what we want to say in our presentation to the transport across the plasma membrane. The two things with the channels and the transporters and the water going through. Then mediated, non-mediated, and active and passive. And what is the difference here? We say here active, there's primary and secondary active transport, and just active transport. Was there now the passive transport? In passive transport, a substance moves down its concentration gradient across the membrane using only its own kinetic energy of motion. Was it the water? Maybe not. And then the vesicles, the 4 or 5 different types of vesicles, we make here a little overview. Osmosis, yes, we don't really understand it completely, but osmosis, you need one substance goes off through the other side because there's more of, it wants to be on both sides the same amount of itself. And that creates an osmotic pressure if there's something on one side which is not part of itself. The amount of pressure that it has because it is going over is the osmotic pressure or something. But I don't really understand it. Osmosis, diffusion, when you have diffusion through the lipid bilayer, diffusion through the membrane channels, facilitated diffusion, active transport, primary and secondary. Endocytosis when receptor mediated, phagocytosis, pinocytosis, and exocytosis. Okay. If you go here for cytology, you can go directly to the next part. we're going to go on to the next thing, and we try to work through all of these individual little things. We already read it in the previous part, all these pages, and try to go over all these individual pages and try now individually to understand each individually. If you were here for cytology, you can go directly to the next part. If you stay here and participate in the leg injury prevention training, then it's very young. Have a good standing leg, everything comes in front and to the side. And the other leg in front and side. And one leg in front and back. Other one in front and back. One leg up, hands out, go over to one side with hands, and to the other side. Middle to one side again, the knee over to the other one. Change. Middle, down, and again up. Over to one side. And to the other one. Middle, over to one side. And another one. Middle, and down. And now, one leg out, and pull the toes in. Up and pull down. Down, up. Other leg, out. And one leg out, pull the toes in, move the foot from left to right. Other leg out, pull the toes in, right, left. And one leg up, press up and to side. Other leg, up and to side. And stand good on. One leg, turn over to the leg. Down into the knee, control slowly over to the other side. And up. Change. Over to the leg. Down. Control slowly over. And up. Stand good and reach always with the heel of one leg to imaginary 90-minute intervals of an imaginary dial of a clock. And with the other leg. 12 o'clock, 1:30 and 3, 10:30 and 9 o'clock, 7:30, 6 o'clock, 4:30 and 3 o'clock. One heel onto the ground, down to it. If you're pregnant, lift the heel off the ground and place it again onto the ground. Other leg. And one leg up and down. The other leg up and down. And stand good, one leg in and out. And now if you like and can, with the whole body. And then if you like and can, the whole swing. And change the leg, in and out. And now the whole body. And then swing. And the next two sides, put your finger down. And out, down. And up. Out, down. In and close the legs. And again out. That was leg injury prevention training. I thank you very much, and until next time.